Hi, everyone. Thank you for joining today's webinar, which will discuss lessons in innovation and biotech from the International Space Station. My name is Amirah Idrus. I'm an editor at Fierce Biotech, and I'll be moderating the discussion. Today, we'll hear from Serena Auñon-Chancellor, a NASA astronaut and medical doctor, Yusuf co-founder and CEO at Kernel Biologics, David Brady, Associate Program Scientist at NASA's International Space Station Program, and Christine Kretz, Vice President of Programs and Partnerships on behalf of the International Space Station U.S. National Laboratory. Please note we'll be taking audience questions at the end, so please be sure to submit your questions via the Slido link in the chat function. We'll start with the panel discussion with Yusuf, Christine, and David. And so we'll begin with the environment. David, what is unique about the space-based microgravity environment of the ISS for research purposes?
Well, Amirah, I'd like to thank you, Amirah, I'm sorry. I'd like to thank you for hosting this event today, first of all, and thanks to LabCentral and also to our other panelists that are participating here. When we start talking about the environment of space station, I like to go back to the old real estate adage that talks about what are the three things that determine the value of any property. And I think everybody knows that's location, location, location. And that's really what it is with the International Space Station as well. And not only is it location, 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 there's really three elements that we talk about on a regular basis as to why using the International Space Station is so useful potentially to researchers, tech developers, and other folks. The first one is, as you mentioned before, is the microgravity element of that. Everything on Earth developed under the influence of gravity. And when you remove that, you actually get to see the other factors that are involved in there and understand your system, your process, your product, whatever it is, much better. If you want to do that in a short duration environment, and there's a lot of ways to do that, and we encourage researchers to investigate these, to see if they will help. There are drop towers, there are sounding rockets, there are parabolic flights that we do on aircraft as well to get short duration microgravity. But if what you're looking for is long duration microgravity for your research, for your technology development, the International Space Station is where it's at. The second element that we talk about, and it has to do with the environment as well, is the ability to test in extreme environments for an extended period of time. Things like a hard vacuum, studying the degradation effects of atomic oxygen, for example, for material science, samples and things like that. Radiation and thermal extremes are some things that you can get on the International Space Station. It's very difficult to sustain anywhere else. And finally, I like to talk about the vantage point, and that's specific primarily to what I call looking up and looking down. It's for Earth science and for space science. Not only is that valuable from the standpoint of the vantage point, but the fact that the infrastructure is there, the transportation system to take your payload up and even down if you need to, the infrastructure on board to provide power, data, video, and anything you need to make your project successful is all there. So I encourage folks to look at this orbiting laboratory and see what we can do to make your project successful.
Thank you, David, for that great introduction. So now we have an idea of, what the environment is up there. Maybe Christine, you'd like to start on why researchers would want to then send their life science research to the space station.
Sure, thanks, Amirah. I think that environment that David outlined creates some things that your results will vary, and they'll vary from what you see in your normal lab on Earth. And with those kinds of changes, you may have different results or create a different product, maybe a different quality of product. So examples are helpful. An example is that things that would grow flat in a dish in your lab because of gravity now don't necessarily form that flat shape and can just grow in a microgravity in a way that may be more open for cell growth. And that could be used, for example, in organoids or tissue growth that bioprinters are trying on the station, printing cardiac cells and other human tissue cells that could potentially then be a regenerative medicine product. So what if they can print something that's new cardiac tissue and then bring it back down and replace and implant it in a patient? Cells grow differently and crystals grow differently. So we have partnerships with pharmaceutical companies, large and small, because the crystal growth is different. and they're trying to find ways that can help. GlaxoSmithKline is an example. They have a project going up on our next flight, and they're going to be working with some of those kinds of experiments in space. And previously, Eli Lilly and Merck have done that kind of work. So it really opens up a lot of opportunities in tissues, in crystal growth, in life sciences and pharmaceuticals. And additionally, there's been some really interesting projects in tissue chips, so chips in space with NCATS and the NIH. And they've got another effort that's coming up soon on one of our next launches. But let me have Yusuf talk a little bit about his specific project, because he too has a project going up on the next launch. So Yusuf, why don't you talk a little bit about yours specifically?
Thank you, Christine. Absolutely. Before I answer this question, I'd actually like to provide some background information about what at Kernel we do. We create messenger RNA therapies that instruct specific cells in the body on how to make their own medicine. Our messenger RNA therapies for cancer are designed to only kill cancer cells while sparing normal ones. This cancer cell selectivity is of utmost importance to us, and we achieve this by taking advantage of how cancer cells normally read the message in messenger RNAs. And they do so, we've discovered, in a completely different way. That's because these cancer cells have unique and structurally and functionally altered ribosomes. These are tiny biological machinery that are responsible for reading the message and producing the proteins in the body. Previous investigations on astronauts showed that they don't have, the crew members that have taken the space flight don't have higher incidence of cancer despite being exposed to high ionizing radiation, which is a known risk factor for a number of malignancies, for a number of cancer types. In addition, recent studies suggest that ribosomes, which are, again, the tiny machinery that are responsible for churning proteins based on the message and the messenger RNA, are significantly altered in microgravity. So with this upcoming study on NASA's NG-14 mission, we plan to test our cancer cell-specific messenger RNAs under microgravity conditions of the space station, with the ultimate goal of developing better messenger RNA therapies for cancer patients that are resistant to leakiness. That's all from Kernel.
Thank you, Yusuf, and thank you also, Christine, for providing so many good examples. And so moving on, I'd like to ask Christine again, how does NASA and the ISS National Lab work with partners like Kernel Bio to utilize the space station?
Well, thanks, because we're always looking for new partners. So with your audience here, we're interested in what they're working on and whether we can be part of their research. One way is, I had mentioned NIH as a group that puts out RFPs through our organization. We work with them on specific kinds of things. NCATS is one of the tissue chip efforts, and they put out an RFP with us, and we collect research and select projects through that. Another is the way that Yusuf started working with us is through a partnership with Boeing and Mass Challenge. And Mass Challenge, based right there in Cambridge, has an opportunity as an accelerator to find really great startups, really interesting projects. And we come in and talk about how microgravity in space might be a factor. And then people who have projects that may have an opportunity to work in space differently and may find that research exciting, put their proposals through us during the Mass Challenge event, and they're selected. And then those proposals are eventually flown in various, and like Yusuf is coming up really soon. But we also take unsolicited project requests. So you don't have to come in through a Mass Challenge or an NCATS or an RFP process, but you can instead approach us and say, I have an idea for a project. What we supply is the up mass, the ride up, the ride back to bring your project back. We have people who can help you to understand how best to do that in space, because it doesn't work the same in space as it does in your lab. And also the use of astronauts. And I know that you're going to be speaking to Serena, and she can talk about her experience doing the research. So those are things that NASA shares with the National Lab. We select projects. And I will say that it's pretty simple to get started. You go to our website, you make a request that you're interested, and we'll send you the package that says how do you put your proposal together, and one of my team will be happy to work with you. And you know what we always say is there's a form for that. So there is a form, and it's a 2-pager to get started, and that's all you need to do. So we try to make it really simple to start working with us on projects that you think might take advantage from being in space.
Thank you, Christine. And so now we know how partners can get in touch. David, could you talk about what kinds of research facilities are available to potential investigators?
Oh, this is such a tough question, and it's tough not because I don't have much of an answer. It's tough to make it concise. So let me start with a resource so that our audience can go and get more information for their specific area, and then I'll try and quickly cover all the different areas that I think would be potentially useful to them. At www.nasa.gov/ISS, as in International Space Station -science. Again, that's www.nasa.gov/ISS-science. And that'll show you, that'll give you a link to all of our facilities, and you can see everything down to the facility level. As a preface to this, I will say there are 6 different areas. We categorize our research and activities on space station based upon the decadal surveys that the National Research Council conducts every 10 years. And under those different categories, we have areas of human research, bio and biotech, biology and biotech, physical science, earth and space science, technology development, and finally education. And for the facilities associated with that, a lot of them are typical to an Earth-based, a terrestrial-based lab, but just to cover them real quickly for our audience, we have typical things like microscopes, glove boxes, cold storage area, centrifuges. And yes, I'm reading from a list because there's so many that I wanted to make sure our audience at least gets a taste of what we have. We also have some small self-contained micro labs, specifically the implementation partners that Christine mentioned, manage a lot of those where you can fly something that may be the size of a CubeSat, for example, and do telescience in there that's mostly autonomous. Some of the discipline-specific things you will see, there are 3D printers. Christine alluded to that somewhat, both organic and inorganic printers. We have plant science habitats. We have exercise equipment for those who are interested in human research. We have free-flying robotics platforms in the event that you're looking at robotic algorithms and want to study that in microgravity. We have a Bose-Einstein condensate facility that a lot of people know. It's called Atom Lab. We have both electromagnetic and electrostatic levitation facilities. We have a combustion facility. We have a granular material investigation facility. We do animal models in our rodent research facilities. And finally, I'll just mention that we have a window observational research facility, a big window in the lab that's very good optic quality. In addition to that, I'll close by talking just a little bit about our external platforms for environmental exposure that I mentioned before, for Earth and space science, for small satellite deploys, and in addition to that, we have airlocks that support the ingress and egress of those experiments. So as you can see, the list is pretty long. We can cover pretty much anyone's research. So please let us know if you need more information. That hyperlink I gave up front is the handiest reference for getting comprehensive information.
Thank you, David. So going from the big long list that David just gave us, we're going to zero in on one company's experience. So Yusuf, could you talk about what facilities Kernel will be using on the ISS?
Absolutely. We will be shipping some human cancer cell line and a human normal cell line to the International Space Station, along with our cancer cell specific messenger RNAs that are formulated with lipid nanoparticles. These cells will be grown in a stable incubator inside the plates that our partners at Bioserve have provided us with. These plates are actually fully sealed, unlike our Earth-based plates. Here is a sample I have here. This is our LabCentral plate that we typically use for experiments here on Earth. It's open top. You can just pipette any liquid inside. Nothing floats, everything is pulled down by gravity, so things are pretty simple. However, when it comes to doing experiments in the space station, because it's under microgravity, everything potentially could float, so you have to take that into account. And our partners at Bioserve have designed these nice plates that are fully sealed, and you go in from the top into a special hole that is covered with a plug using a syringe fitted with an appropriate size needle to ensure that the liquids that you work with for cell culture don't go all over the place. And there's a special incubator called Sable Incubator on the space station that we will be utilizing. This is a modified cell culture incubator that is very similar to, in terms of its function and capabilities, what we typically work with here on Earth. It maintains a proper temperature and carbon dioxide level to enable cells to grow effectively. And our cells will be treated with our messenger RNA drugs in a glove box inside the space station. This ensures that the reagents don't go outside an enclosed space for biosafety of the crew members. And they will be placed back into the incubator, and 24 hours later, the plates will be brought to the NanoRacks plate reader on the space station for functional readouts. And one of the plates will be returned to us for additional testing with the SpaceX 21 mission. We will be performing additional on-Earth testing on that final plate.
Thank you, Yusuf. And we'll pivot a little bit. So my next question is about the recent launch from SpaceX that launched American astronauts from American soil to the space station for the first time since 2011. So maybe, David, you could start, but what does that recent launch mean for our future in space, and what does the future look like on the space station?
Well, we're very excited about the new commercial crew program capabilities that are brought forward by our commercial partners, SpaceX and Boeing. And as our administrator has said a number of times, we are trying to make it where NASA is one of many customers in low Earth orbit, and that the private industry will have a market where they can build capabilities in low Earth orbit that both will serve terrestrial economies, as well as allowing us to continue out into the cosmos and push humankind out into the cosmos. One of the most obvious things that happens is that based upon the size of the vehicles and the capacity of the vehicles, we're able to carry more cargo and more crew up. So we've typically been operating the space station for about the last 20 years or so with a crew complement of about 3 crew members operating what we call the U.S. operating segment, which consists of the American portfolio of research, the Japanese, the Canadians, and the Europeans as well. So we're going to go from 3 to 4 USOS crew members, which will allow us to double the time that we're spending on research. And the reason is that it takes so much time to maintain the station on its systems, things you see like spacewalks and things like that, that when you add an additional crew member and you can devote them almost 100% to research, it's going to double our time that we're going to be able to spend on research. But in addition to that, and more importantly for people like life science researchers, is that instead of going up on a Russian vehicle that has its own schedule constraints associated with the way the Russian Space Agency operates their program, we'll be launching instead from American soil. And I know a lot of people think that that's just a patriotic diatribe, but the reality is that when you bring the American research point of departure closer to the research labs in the US or wherever, and you bring it back to near American soil, then you get that scientific benefit of having, especially on life science samples, which are a lot of times degrade over time and have to be kept cold. Then you have an additional benefit on being able to get them there and not spend as much time out of the lab environment, get them on station, do your research, and then also bring them back. It also gives us additional schedule flexibility on when we bring them back, which could be very useful to a researcher who wants to vary the length of time between departure and return on that as well. We also pick up more, as I mentioned before, more cargo mass. So instead of being able to fly maybe tens of pounds worth of research, it'll be like hundreds of pounds instead. So this year is a little different than others in that we're transitioning to this new capability. But this capability has so much potential growth for the future. We are extremely excited to see it get off the ground with the test flight earlier this year and the first operational flight coming up end of next month.
Thank you. Christine, would you like to jump in?
Sure, we're excited about it too. What's happening at the National Lab is that we've grown the number of payloads over the years, so that we're into about 90 payloads per year and more coming all the time. So we need more capacity, and that is capacity of great researchers like Yusuf and others who are either working for a smaller company or a university or whatever, the more payloads and the more astronaut time we can get, the more we can put on those rides up. Even more importantly, thinking into the future, if you watch the SpaceX launch with Bob and Doug, our astronaut dads, as they sometimes are called, it was terrific footage from NASA and really great during the pandemic to be able to get such insight. But you'll notice that there were 4 seats in that capsule. I like to say one of them's mine, but one of them could be, maybe David, one of them could be Yusuf. So there could be a time when researchers are going up to the space station and not just sending through BioServe or a partner, but going themselves and working alongside the astronauts. It's not that far off, and there are already organizations working on the training required to put Dr. Urkel into space and to do that kind of research with them. Additionally, NASA signed an agreement with XIII as one of the partners that will be doing some additions to the space station. And when you talk to Serena, she'll tell you it's snug up there. More room could be useful, more space, more room for more kinds of facilities. David listed a bunch, but you can always think of the next new thing that you need. So XIII is going to be adding a new node on with some more sleeping quarters for astronauts like Dr. Urkel to go up and stay there, but also more facilities. And eventually, the ability to manufacture in space is going to be possible. So as these sorts of bio work where you might need to make something with organ tissue are going on, the requirement to make that as a product and be able to bring it back down is going to be required, and that will mean more room on the station. So we're excited about what NASA is adding from a commercial standpoint, not just the rockets, not just SpaceX and Boeing, but other components that will add to the capabilities on the station.
Thank you, Christine. And since you mentioned the pandemic a little bit earlier in your answer, I'll ask the next question, which is how has NASA and its partners reacted to COVID? I don't know, David, would you like to start, and then Christine?
Sure. Yeah, we take great pride in being sensitive and responsive to our researchers' needs. And the thing is that, like all organizations, businesses, and groups across the globe now, we're having to adapt on how we do our processes, how we do business. We do a lot more online and a lot more virtual items. But one thing that hasn't changed is that we've always had a philosophy of being putting our clients' need, our research clients' needs first. Specifically, what I'm talking about is that we have a launch schedule where we send up cargo vehicles on the order 4 or 5 times a year, and we send up crews about 4 times a year. And as I mentioned before with commercial crew, we're looking to adapt our schedule to meet our researchers' needs in addition to our needs to change out our astronaut crews. But one thing that the pandemic has done for some of our researchers, it's impacted their ability to prepare for flight. But we've always been an organization that says, hey, we're here and ready to go whenever you're ready to go. And we also have quite a few researchers that are waiting to fly. So it's usually not a problem for us to change our flight schedules and be more flexible, and then just move some folks back if they need a little more time as a result of the pandemic. I just wanted to mention that I think over my 3-decade career with NASA, one of the things I'm most proud of is that we have gone from a largely insular agency who was very proud of their work and did very good work, very impactful work with the space shuttle in the late 80s and early 90s, to being willing and interested in collaborating in just about any way we can under US law. And because of that, we've not only created new collaborative relationships with international partners, but also with commercial partners and research partners. And we'll continue to do that. Like I say, our goal within a pandemic or outside of it is to be responsive to the needs of our researchers and provide the platform they need to get their project, make their project successful.
And Christine?
Sure. I would say there's also some research that could affect how we handle viruses. There's vaccine research that has been done in the past and some lined up for the future. The note there is that it doesn't solve the immediate situation, right? So if we're doing vaccine research with one of the pharmaceutical companies in space, you have to add extra time on because there are only so many rides up and back every year. So it's not something that helps us with our current emergency in the COVID-19 pandemic, but it certainly can add value as vaccines are required in the future for opportunities that these could be made in space. They could be made differently. We could have insights from the vaccine research that is going on the station even now. So I think there are opportunities, but space does take time. And so we won't see anything in the immediate COVID situation from space station.
Thank you, Christine. And I'd like to finish out with Yusuf, specifically Kernel's experience prepping a payload that will launch to the space station and how that experience may have been affected by COVID.
Sure. Well, prepping a payload, especially using a novel cell culture system like we did with our colleagues at Bioserve, these 12-well systems that Kernel is using for the first time, usually takes a while because you have to optimize everything and you have to do a lot of troubleshooting. Bioserve is our implementation partner for this study, and they manufacture these plates and they trained us on how to use them. One of their team members, Carla Hahn, has warned us at the beginning of this project that things typically take a lot of time and a lot of optimization, but we always triumph. That was the words she used, and that has been exactly the case. We did have to do a number of experiments. We did have to do a number of unforeseen troubleshooting, but we eventually overcame all those difficulties. And this was, in a way, NASA's own historical record that they never give up. They always succeed. You sort of rise up to the occasion and do your best in handling all the challenges, especially technical challenges that might emerge. With Bioserve team's help, we have performed a number of optimizations, for example, for cell seeding, for culture temperature and duration, as well as media improvements to ensure that these cells can grow up to 9 days in cell culture. Just for reference, typically on Earth, we would do experiments where every 2 or 3 days we would replace the medium for these cells to make sure that they're happy. Whereas with this project, for the first time, we're going up to 9 days while continuing to grow the cancer cells. And the cells will be seeded at LabCentral, and then we will be placing them on a plate habitat. This is a special container that Bioserve has designed that allows the cells to be maintained at proper temperature and CO2 levels. Then for the launch, we will be shipping them to the launch facility in Virginia, the Mid-Atlantic Regional Spaceport. 36 hours before launch, we will be doing the transfer to the crew members. And our work at LabCentral during COVID has continued despite the pandemic. I want to say thanks to all the great staff and the administration here that has put in place a number of preventive and protective measures to allow us to work safely, but also effectively throughout this pandemic. I also want to thank Governor Baker for his special treatment of the biotech sector, especially during COVID, especially of companies who are working on essential life-saving medicine. They have continued to be able to do their work effectively. However, we did have some step backs early on. We had to make sure that everything that we do was safe and everything that we do was not being rushed through. Before the launch, NASA requires us to actually perform the entire experiment here on Earth, just like we would do on the space station. And this study was performed for Kernel in July at LabCentral, and we got a special permission from LabCentral for a Saturday all-day event where a team member, Ms. Shankini from Bioserve team, came to LabCentral, where there was almost nobody else in the building except for Kernel team, and we went through the entire experimental validation testing protocol. This EVT is required by NASA, and Ms. Shankini provided us with valuable feedback, and we streamlined our protocols based on that feedback. So LabCentral has shown us great flexibility in making sure that we can find ways to work around all the difficulties that COVID presents. And overall, I would say, while this has been sometimes challenging, overall, it was highly rewarding. It's a great experience. I want to say big thanks to my colleagues, Burak and Jaffer, for their incredible work that they did at Kernel, and to our advisors at Harvard, MIT, and the biotech industry. Also want to thank the ISS National Lab and Boeing for sponsoring the study through Mass Challenge. Kernel has received awards from Amgen as well. Also big thanks to Amgen and Y Combinator, who have been very supportive to Kernel's drug development efforts with computational biology pipelines. Together with all these organizations' support, we look forward to launching our messenger RNAs to space in 2 weeks with NG14, and using the data we get from that study to develop safer and more effective medicine for cancer patients.
Thank you, Yusuf. And I'd like to thank Yusuf, Christine, and David for such a great discussion. We'll hear now from NASA astronaut Serena Auñon-Chancellor. And a reminder to the audience that we will continue to collect questions from you. Please be sure to submit them via the Slido link in the chat function. And right after we talk to Serena, we will get to your questions. Oh, there you are. Hi, Serena.
Hey, how are you?
I'm great. How are you doing?
Very happy to be here.
Perfect. My first question for you is a very big and open-ended question, so I'm just going to ask the first two together. The first question was, what was it like working in space, which is, like I said, very big and open. So the second one is, what were some of the most memorable experiments or projects that you worked on while you were at the space station?
Yeah, so let's start with your first question. That's easy for me to answer. It's awesome. You know, at first, when you first get up there, you want to keep that kind of Earth, kind of 2D, feet on the ground, body positioning, and then you very quickly learn that you can work in 3D up there all over the space station, and you can perform science all over the space station, on the ceiling, on the floor, proverbial floor, on the walls. And so it's really kind of cool to watch your body learn to live and work more efficiently in microgravity. You're just very ungraceful at first, and then shift to, I don't want to say ballerina, but a graceful science ballerina. You know, certainly by the middle of your stay at least. So, you know, some of the coolest science, I have to say, that I worked on during my 197 days was science. It was medical science, science that impacted the lives of people on Earth. And it's important to me because I'm still a practicing physician. I was actually in the hospital earlier today. And this is what people care about. People have a personal connection to space and the space station and NASA when they know that a lot of the work we do is really trying to impact their health down here. And so I think people are kind of shocked to learn that we don't get briefed on a lot of the science before we head up there. Some of the more complicated experiments we do, but I really learn about it once I get on orbit. And when I began to open up and look and read about some of the science that we were doing, I was stunned. I mean, it was fantastic. I worked on, you know, looking at cancer chemotherapy, how the endothelial cells, or cells that line our blood vessels, how those grow in microgravity. We looked at Parkinson's disease. We looked at how muscles atrophy. And this is important because patients, when they're in the hospital, are in bed rest for a long period of time, and your muscles change, your tendons change, and the way you rehabilitate changes. There was so much that we did up there with regards to human health. It was stunning to me as a physician. I was pleasantly stunned. I mean, it was really neat to be able to open up each of those packages and read about all the information about the experiment beforehand. And it's still what I love talking about down here, not only with all my scientists and colleagues and astronaut friends, but my patients. They love to hear about it. They love to find out what NASA is doing for them on board the space station.
Yeah, thank you. And you talked about a few different challenges being up there. One, getting used to microgravity to begin with. Another, the fact that you kind of just brief yourself on the science when you're up there. My next question is, was there anything you experienced that was maybe harder than you thought it would be? Or on the flip side, if there's anything you thought would be hard, but actually turned out to be easier?
So I think the harder things you deal with at first is getting the experiment set up. We have several different areas we work in. I worked a lot in the U.S. lab in our microgravity sciences glove box, which is like a big operating glove box. And at first, trying to keep everything down and keeping it from floating away is your most difficult thing. And that's why, you know, scientists like Yusuf are paired with an implementation partner. And so when everything gets packaged and sent up, you literally are able to set it up inside. Everything has a Velcro tab. My toothbrush has a Velcro tab on the space station. I don't let anything float away, let alone the science. So at first it was hard though, just learning how to deal with that. And then again, your brain, your body gets so used to working in space. It did get easier with time. The one thing that was easier to deal with up there. You know, when I was at first, I thought that some of these projects and experiments were complicated, and I certainly didn't want to mess anything up. And if I had questions, but the great part is when we're doing these experiments on orbit, I am talking with the scientists directly on the ground. There's multiple cameras. They can see me and what I'm doing. I can ask questions real time. So as we're doing an experiment or plating, I can say, hey, Yusuf, why am I doing this right now? What if I do it this way? Can we change this around? And they talk to me right then. So to me, that was very, very easy to do. And that scientist could have been in Oregon or Florida or international, but they get tied in. And you mentioned the name Shankini earlier. I don't know if she's listening from BioServe, but I worked with her a lot on a lot of these experiments, and she's fantastic. But it was much easier than I thought to interact with the scientists directly on the research, and it made it more special.
Thank you. And, you talked a lot about acclimating to life on the space station. So how do you acclimate to life back on Earth after spending, 197 days in space?
Yeah, it's a little rough at first. 197 days when you land, you don't feel great. I mean, you're so happy to be home. Certainly that first day, that first 24 to 48 hours is a little rough. Your body, I call it reprogramming. Your brain, which hasn't had to deal with a gravitational stimulus, all of a sudden has gravity, and it is frantically reprogramming to say, oh my goodness, well now if you tell me to stand on one leg, I've got to remember how to do that. And so the brain is sending all these signals. The biomechanics of movement, in a sense, have kind of been dormant. They've been asleep, and it's got to wake back up. And so it was fascinating, as bad as you feel while you're undergoing this rehab, it is fascinating to watch the brain and the body wake up all those pathways, those neural pathways, and be able to move just like you did prior to launch. It takes time for your body to recover. Everybody's different. And I have to be honest, my first run, when I tried to go running outside, I felt like I had a gorilla on my back. I was not, it just didn't feel normal, but it doesn't take long before you get your Earth legs back again.
Yeah, that was taking my next question out of my mouth. I was thinking, when you talked about like reprogramming and all of this, I wanted to know, like, what does it feel? So does it feel like you have a gorilla on you for all your activities, I guess?
And, initially when you're getting your rehabilitation with our strength and training specialists and our flight surgeons, they ask you to, I remember they asked me to just stand about 6 feet back from a wall and squat and throw a ball against a wall and catch it. And I thought, well, that's simple. That's one of the easiest things in the world. And it nearly hit me in the face because I wasn't used to having to raise my hands up and the way my body was positioned. But by the second time or third time, it was back to normal. So the body remembers. The body is fascinating. It is resilient, and it remembers.
Thank you. And since we're talking about resilience, what are some parallels that you can draw, between the time on the space station and to quarantine during COVID?
It was interesting. So quarantining, I was lucky in the sense that I was not locked in at home. So I was actually working in the hospital during COVID-19 on a COVID ward. So in the hospitals themselves, we were very concerned about amount of the PPE that you see people wear, the masks and gloves, and consumables were certainly something that we kept track of every day. And that really reminded me of Space Station. Space Station, we don't have an unlimited supply of anything up there. We're very careful with how we use everything, when we use things. We don't have an unlimited supply of test tubes or needles to draw blood or even bandages. And so it was very easy for me in the hospital during the time of COVID to immediately think of the space station, because in the space station, any medicine that we have to perform is almost medicine in an extreme environment. And COVID was the same. It was a disease we had absolutely no knowledge about. We had limited supplies, and we were just doing the best we can. So to me, I know other people can't draw that parallel as easily, but to me, it was like being on ISS.
Thank you. And my last question was, how have you coped with the physical distancing back here during COVID?
Yeah, so physical distancing, it's even here in the hospital. I'm in my office building right now, which is full of a bunch of doctors. We are all, when I'm not in my office, I am wearing a mask. So we are always being safe. We are maintaining our distance. I think it's almost become a little bit of a way of life where you just maintain that spacing. I will say I miss seeing people's smiles. I miss seeing people's faces because it just makes it more personal. And that's one thing that's been a little more difficult to deal with during COVID. And I will say that when you're on board the space station, boy, besides the folks you're with, you are as physically distanced as you can be from the rest of the people on this planet. So they're probably the safest up there.
Thanks. And could you talk a little bit more about working in the hospital during COVID? I know that's not the typical experience for a lot of people. A lot of us have been kind of stuck in our homes.
It was very hard. So I worked in the early part of April. I'm currently in Baton Rouge, Louisiana, and the state got hit extremely hard, as many states did, by COVID. And we had a lot of patients in the hospital that were very sick. And, I think it was difficult because their families could see them, but only under very limited circumstances. It wasn't the same as, when hospital was operating in normal times when your whole family could come in the door. We were really trying to prevent as many people from getting infected as possible. So we were the patient's family. I'm proud of that. It was also one of the hardest things I've ever done. Again, it's a disease process we are still learning about. No one has the answers. I don't care what they tell you. Our treatments are evolving. But viruses are scary because viruses are tiny, tiny, tiny particles, and they don't care if you believe in them or not, and they don't care if people choose to gather or not. They're always there. And so we're just trying to do our best to keep people safe, keep our most vulnerable population safe, certainly our elderly, even our youngest, or people with compromising diseases where their immune system's not working well. And I think we're getting there. We're going to get there towards a vaccine. And I won't say it's commonplace now to see COVID in the hospital. We have it every day. We're still very careful. But it certainly wasn't as bad as it was back in April.
Thank you, Serena. We do have a couple more minutes before I have to start the Q&A. I don't know if there's anything particularly memorable, either from your time on the space station or even, back here working in the hospital during the time of COVID, that you'd like to share with the audience before I get to questions.
I always just, again, one of my big messages when I go out there and talk with folks about science on board ISS is that I want them to realize, and I remember hearing Christine say this during one of her talks too, is that the majority of life science research we do up there is for you. It's for people down here. It's not for me to get to Mars. It's not for astronaut Joe to get to Mars, although we do that research. It's for folks down here. And so that's why there's several clinics I work at, even some free clinics still in Galveston, Texas. And they ask me all the time, tell me about that, doc, tell me about that Parkinson's research that y'all are doing up there. Are you going to help us better? Are you going to be able to help us find medications to better treat it? My answer is yes. And they take such comfort in that, and they should, because the ISS, like every other research institution or academic institution, is doing its part. I'm not saying we're the end all be all and we're going to be the ones to find a cure, but everybody has to play a part, and every little part we play sheds more information, new discoveries. And let me tell you, no one else can do the research that we can do. Before we started this Q&A, I was talking with Yusuf. I wanted to hear more about his research, and it's fascinating. So I love talking with scientists. I love to hear about ideas and connecting people. And so I love doing webinars like this because I can't see the audience. I know you're out there, and I hope to hear your questions. But I hope you took something away from this today, because I always do.
Thank you so much for sharing that, Serena. I think we're going to try to get the rest of the panelists back on the screen for the audience Q&A. And I see we're getting them one by one. So I think I'll just go ahead and start the first question, and we'll see who should field that one. The question is there any type of experiment or research that we're not already doing in space that you would love for NASA to do one day?
Tough one. I think that we're doing so many different kinds of research that I don't think I've heard of anything that we're not able to accommodate. And what I've noticed in just the 2 years I've been with the National Lab, the work gets more and more complicated, making it more of a challenge for NASA. It makes it more of a challenge for the astronauts. It makes it more of a challenge for our research partners and the implementation partners. But I think that it's been noted, we rise to the challenge, our partners rise to the challenge, the researchers provide us all the tools that we need to help them. And so I haven't run across anything that we're not working on yet, but if your audience thinks of something that we haven't tried, bring it on.
Yeah, if I may add.
Yeah, go ahead.
Yeah, I agree with Christine, because the thing is that I can't think of anything, but there's a number of things both in scientific discoveries that we found from doing the research on space station that we didn't expect. And there were some capabilities, for example, the ability to launch small sats that we never thought about using to the level that we have used off station. So don't think that we've thought about it all. Come on, please bring your ideas, because you may be the one to come up with a new way of using this platform that we haven't considered yet.
Thanks. The next question is, what are the criteria used to determine if a company can send their research to the ISS? Like, which are the deciding factors or limits?
I can take that one too, and Dave, you can fill in after me. Generally speaking, they're U.S.-based organizations or have a U.S. affiliate. So you may even hear of us working with Idrus, but they have a U.S. presence, and that's the team that we're working with, plus other universities and small startups. We like to stick in areas that are our focus areas. Life science is a big part of that, but we also work in advanced materials, in space production opportunities, and generally basic science that gives us new insights. So it does have to be real science. It has to be something that hasn't been done a million times before. If it's something that has been done, we'll point you to publications in that area. And then last but not least, it's a matter of it cannot be so large that we cannot put it on a rocket. So if it's bigger than a refrigerator, it's probably a little too big for us to handle. But David, are there any other things that you would suggest that are parameters?
No, I was just going to mention that the URL I gave earlier, the hyperlink I gave earlier, goes to our research and technology page. And on the left-hand side, there's a hyperlink there, and it says opportunities. And so you can click on that. Again, that was www.nasa.gov/ISS-science. And you can look for research opportunities there. We've also recently, in the last year, NASA has started soliciting opportunities for doing commercial for-profit ventures on space station. It's just a small part of our budget, but it is part where we're trying to grow a low Earth orbit economy and encourage commercial interest, private interest to get involved in that. So if you have something that's for-profit, don't think that you can't do it. You know, come and ask us about it. We have a helpline also on that same website that'll answer your questions.
Thank you. Next question is about gravity. So is there a difference between gravity versus centrifugal force from the test subject in the ISS? I don't know who that is a question for.
So are they asking?
Is there a difference between gravity, like the external force from a celestial body, that's part of the question, versus the centrifugal force, which I'm assuming is to do with the space station?
I can try that if you want.
Go for it.
Yeah. Yes, there is a difference between that. The astronauts are in what we call free fall. They're essentially matching. They're going so fast around the Earth that they're falling towards Earth, but they're going so fast they never hit it. So the gravitational force still exists on board the space station, but it's negated by the constant free fall of the space station. And that's why they float in a condition we call microgravity. Whereas we have some experiments, for example, some fluids in tanks, fluid dynamic experiments, where you'll see videos of the astronauts spinning it around to check out the slosh characteristics and the settling characteristics. And that's when we take advantage of using centrifugal force in the microgravity field. So those are the differences right there. And I hope that is a good enough example. Serena, I don't know.
No, now I understand the question. And what's kind of cool, what David said was we also have a small mini centrifuge on board ISS. One of the cooler experiments we did looked at cement solidification and how cement basically forms in differing gravity conditions. And why would you want to know that? Well, if you want to build a lunar habitat someday or a Martian habitat, I doubt you're going to have to create that cement when you go up there. And how do the fluid characteristics change? And is the structure of the cement itself, the internal architecture, is that the same? It was fascinating, but we put several samples in a mini centrifuge and then sent that all back down on the ground for folks to take a look at that. So it's pretty cool.
Thank you both. I will admit I also didn't understand the question, so thank you for elucidating on both fronts. Next one is for Serena. What made you want to become an astronaut?
Oh boy. Well, probably since I was very little. I can remember 8 or 9 years old, and it was mostly just because I watched shuttles launch over and over and thought it was the most fascinating thing ever. And so I remember my father and my mother sitting down with me saying, "Do you want to work for NASA someday?" I said, "Yeah, I don't know how." And my dad goes, "Well, I'm an engineer. You should be an engineer." So my undergraduate education was in electrical engineering at the George Washington University. And so, and then I went to medical school after that. So what I tell folks is, man, folks of all types of backgrounds work at NASA in many different fields. So I love being a physician. I love being an astronaut. So it was a perfect combination.
Thanks, Serena. And this one might be one for Christine. This person wants to know if there's any research going on the ISS regarding the effects of microgravity on 3D printed tissue.
Yes, there is. So the bioprinter is doing that. It's the heart tissue specifically. The company that is doing that work is called TechShot, and their bioprinter is called BFF. And so if you want to see more about what's going on with BFF, and you should try the TechShot website, or there are a number of things on the International Space Station National Lab's website. That ISS 360 is our blog, and we have a number of videos, which you'll be able to see some of the work there on the BFF.
Thank you. There's another question for Serena. Do astronauts celebrate holidays in space? I imagine that would be difficult being away from your family.
We do celebrate all holidays. We celebrate all birthdays, all holidays, both American, Russian, European, you name it, whoever's on the space station. One of my colleagues that I flew with, Oleg Artemiev, we celebrated his daughter's 7th birthday, 7th-month birthday, 8th-month birthday, 9th-month birthday, 10th. It was a good excuse for the crew to gather and just celebrate family. And it is hard being away from your family. I was up there during Thanksgiving, so I try to do all the things that I normally did on Thanksgiving Day, which was watch football, watch parades, and we actually had a Thanksgiving dinner up there. Not quite like home, but it comes pretty close. So yes, holidays are absolutely big up there when you're exploring and you're away and off the planet. It's important to keep those things special to the crew, and it's important that the crew spends time together to reflect on them.
Thank you. Here's another question about research in microgravity. What do we know about how stem cells behave in microgravity, and is there any research going on about that on the ISS?
I'm going to try this, and then I think, Serena, you did some work on did you do some stem cell work while you're up there?
Kate did a lot of it. Peggy did a lot of it. I did not do as much work on the stem cells as I did the endothelial cells.
So there are a number of organizations that are working on stem cells, and regenerative medicine as a general topic is very important, and we think we'll have a lot of results from the microgravity environment. So there are a number of universities involved in a number of organizations that support that research. And I think we're really just at the beginning of it. So we've, just like everything, you start slowly, find some new things, and then get more and more complicated. But there are some organoid projects, for example, that went up, I want to say just about a year ago, and those results are being looked at now. And then what happens in all of these projects is, and the stem cell work is no different. You start with baby steps and you learn what you're going to learn. You get that material back down, as Yusuf will get his information back down. And then what we would expect would happen is from the findings, from those learnings, you put your next experiment together, and maybe a third and maybe a fourth, and each one gets you a little bit more learning. So Yusuf will do this project, and then he'll have findings that will create a next in the next step in the chain.
Thanks, Christine. I just want to remind the audience we have about 7 minutes left for the Q&A, so if you have a burning question, please don't hesitate to submit that via the Slido link. Next question is, how are cells sent to the ISS impacted by supergravity, 5G or more, during the takeoff?
I don't know. Do you think, Yusuf, did you figure this out? I'm going to let you field this one.
This is something that we thought about the main goal of the study was to actually take advantage of the microgravity, and we started thinking, well, if you're going to send us with a rocket launch and the cells are actually in live culture, they will be facing up to 5G or perhaps even 10G during launch. But then we realized the centrifugal forces that we typically use in our labs when we try to spin down our cells for culturing is actually up to 125G.
Wow.
So that's when I stopped worrying about the launch G, because in our routine work here on Earth, we apply much bigger centrifugal forces, and those typically don't result in major changes in cell phenotype or in cell viability. But that being said, I want to connect back to an earlier question about how centrifugal forces differ from microgravity. Microgravity is actually an environment, especially in the space station, where the cells don't perceive almost any gravity. And these centrifugal forces that we apply to cells to actually work with them in the lab through our centrifuges actually exert a very high G force, typically 125, sometimes up to 200 G, that may be affecting some of the research outcomes. So I would urge my colleagues in biomedical research field to consider actually taking deep notes when they do cell centrifuge before culturing the cells, that it might perhaps have some impact on some fine biological processes that involve, for example, translation.
Thank you. And I just have a last couple of questions for Serena. One of them was, how did you feel changing from an electrical engineer to medicine? An engineer is asking.
Oh yeah, it was interesting. So it required a little bit of a change in the way I thought, but at the same time, you know, engineers are critical thinkers, like many of our scientists are. And so it was kind of natural for me. Engineering is very detail oriented, and I'm an internist, so I'm very detail oriented about the body and all the physiologic processes. So it was kind of neat though, because when you work for NASA, NASA to me is a sea of engineering and science with little islands of medicine kind of dotted throughout. And so the human body, which sometimes we call the human system, has to interact with the rest of the vehicle system, and everything's got to work perfectly to protect that human system. So in a sense, you're taking the most important part, a part that's not predictable and always different, and trying to make sure that it interacts well with the ISS space station system and all the scientific systems on board, to ensure that, you know, astronauts are healthy and happy on orbit. So to me, engineering was a perfect lead-in to the field of medicine, especially space medicine.
Thank you. And one more question for you. It is, do you think you'll return to space for a future mission?
Ooh, not anytime soon. I think I landed about 18 months ago. So, I mean, I'm sure my bones are still recovering. It does take time for that bone mineral density to come back. And I'm actually really happy being on the planet right now. It's a gorgeous planet, even with all the hurricanes and everything else. It's beautiful, and I love being here.
Thanks. And we just have a few more minutes. Thank you, everybody, for participating in today's webinar, for the great discussion, and also to all the audience members for tuning in and sending in such great questions. We didn't get to them all, but they were great. Thank you.
Thank you.
Thanks. Take care of yourselves.
Here, everyone.